welcome to the ParAqua-COST, the science podcast that shines a light on the newest and most interesting research being conducted in the fields of aquatic and marine ecology. My name is Joe Money. I'm a PhD student and the host of this podcast. Today, in a podcast first, I'm joined by two guests, Erika Martinez Ruiz, a postdoctoral researcher at the Leibniz Institute of Freshwater Ecology and Inland Fisheries in Berlin, and her recently graduated master's student and research partner, Nele Guttmann. We'll be discussing their research into the impacts of anthropogenic pollution on aquatic ecosystems, and specifically focusing on their recently published paper focused on the chemical damage that cigarette filters can have on algal communities. Erika, Nele, how are you doing?
I'm doing well, thank you.
Doing good. Thank you for having me.
No problem. Thanks for both of you joining me. As I said in the intro, this is the first time we've ever done a podcast with two guests, so we're going to see how it goes. We're going to try our best, and everything's going to be a learning experience. How have you guys been lately? How's work and life been?
It's going well, I would say. For me, it's going okay. It's very cold now here in Berlin, but at least we have a bit of snow, which makes things a bit nicer. And yeah, in work, normal stress and normal enjoyment, let's say.
Nice. I know the cold conditions can be difficult when you're working on a lake, so hopefully less field work at the moment and more lab work.
Totally.
Yeah, for me, it's going pretty well as well. Not too much biology stuff at the moment. I'm still looking for a job. I finished my master thesis last year. But I'm also not enjoying the cold, but I do enjoy the snow. So that's nice. I'm happy that we've got a little bit of a Christmas feeling, very, very belated Christmas feeling in Berlin.
Better late than never, exactly. Yeah.
Exactly.
So could you both give us a quick summary of your background, education, and research history? Why don't we start with you, Erika?
Yeah, sure. So I was born in Mexico, more specifically in Mexico City, and then I studied my bachelor in chemistry, bacteriology, parasitology in the Escuela Nacional de Ciencias Biológicas of the Instituto Politécnico Nacional. At the same university, I did my master's studies in the program of chemical and biological sciences, and since there, I have been studying the effect of anthropogenic pollution on aquatic microorganisms. When I finished my master's, I moved to Berlin to do my PhD at the Technische Universität Berlin. And once I finished my PhD, I wrote a proposal for the DFG. It was accepted. And since then, I have been working at the IGB with this very interesting system of the cyanobacteria and chytrids, and again, with the effect of anthropogenic pollution on aquatic microorganisms.
Nice. That's a very international research history you've got there. I can imagine you're really suffering with the winters here compared to Mexico.
Oh, yeah.
And yourself, Nele?
Well, not so international for me. I was born and raised in Berlin, and I went to a university here as well. I did my bachelor and my master's thesis at the Freie Universität of Berlin. And then I was lucky enough to find Professor Dr. Justina Wolinska's working group at IGB for my bachelor thesis, which is also where I returned to for my master thesis, and this time under the supervision of Erika. And I say lucky because I didn't only get to work on this paper, but I also got to publish a paper with my bachelor thesis just recently. I think it was shortly after this one was published, completely unrelated topic. It was a meta-analysis, was very interesting to work on. So I guess I got very lucky getting into that working group and getting so much like hands-on experience with research. Yeah.
Well, that's really impressive. I'm very impressed if anyone gets a paper from their master's, let alone their bachelor's. So that is very impressive indeed. Definitely lucky to find Erika, but I'm sure that you put in a lot of hard work, and definitely this paper that we're going to talk about, I really enjoyed. So I'm looking forward to discussing it with you.
We both did. We worked pretty hard on that one. Yeah,
Nice.
It was a lot of hours.
Nice. So one of the study species of your joint research is cyanobacteria. Now, we've talked previously on the importance of algae in freshwater and marine ecosystems on this podcast, in fact. But could you summarize to the listeners what cyanobacteria are and perhaps how they differ from phytoplankton and algae?
Yes, sure. So cyanobacteria are indeed part of the phytoplankton. They are a type of bacteria that can do photosynthesis, which means, in very simple words, to transform the light energy into chemical energy, just like plants do. However, unlike the cells in our bodies, cyanobacteria don't have a nucleus, which is a special compartment that holds the genetic information. Instead, the genetic material of cyanobacteria floats freely inside the cells. And this is the main difference between cyanobacteria and algae. So cyanobacteria are not algae. Even though they have been very often called blue-green algae, this term is simply incorrect. So in very simple words, cyanobacteria are bacteria that can make their own food using the light.
That's exactly the point I wanted to touch on, that they are often referred to as blue-green algae in the popular consciousness, but actually aren't algae themselves. Very confusing. Whoever named that definitely deserves to be fired, definitely. But yeah, I think we could definitely come up with a better term these days, right, for cyanobacteria. I think something more popular, better for the public, to say rather than cyanobacteria. It definitely sounds a bit scary. So from my limited work with cyanobacteria, they often seem to be characterized as the evil or bad versions of algae. So maybe you could explain the roles that cyanobacteria play in aquatic and marine ecosystems and perhaps why they are important to both wildlife and humans.
I wouldn't say that they're bad. They're actually really important for the environment since they are photoplanktonic, as Erika mentioned. And phytoplanktonic organisms are responsible for about half of the world's primary productivity. And cyanos can, again, as Erika mentioned, do photosynthesis. So they produce energy and oxygen and make them available to the organisms around them. And this distinguishes them from other bacteria. In that sense, they're really, really important to the environment they live in. And this works super well as long as there is like a healthy balance. And I think the reason for cyanos being so often labeled as evil or bad, as you said, is because they're mainly discussed in the context of toxic blooms. So proliferating cyanobacteria form blooms that can negatively impact the environment. And this is where this like evil headline or this evil stamp comes from, I think.
Yeah, that's definitely how I've seen them reported, before I got into science, that there was a cyanobacterial bloom, there was a blue-green algae outbreak in a lake or something like that, and it was affecting the water quality. So what is it about these blooms that has a negative effect on the water?
So the problem with the blooms, where there are many, many problems that we can associate with blooms, but to start with, once the blooms developed, they can change the color of the water, produced they can change the odor and also the taste of the water, which can directly impact, for instance, all the water bodies that we use for recreational activities, but also the drinking water sources. But apart from that, blooms can produce a massive death of organisms because they once the bloom is dead or the cyanobacteria are dead, they can consume a lot of oxygen, or bacteria that are going to take or eat this cyanobacteria mat that is dead. They can perform a lot of respiration. So they are going to use a lot of oxygen. So they are going to produce some zones that are going to be anoxic. So this can affect all the organisms there. And also one of the main problems associated to cyanobacterial blooms are the ability that some cyanobacteria species have to produce some toxic compounds that we call cyanotoxins that are potentially toxic to humans and other living organisms like animals or plants. And actually, I think it's very common, unfortunately, to see some of the headlines that some dogs, for instance, have died due to poisoning with these toxins
Yeah, it sounds like it can be a real issue. And as Nele said, in ecology, it's always about this balance, right? So it sounds like too much of this and too much of that can really throw the systems and the cycles out of balance. So the specific form of pollution that you have chosen to focus on in this research paper is discarded cigarette butts. Now, I have to say that I really enjoyed reading this paper, and it's a really creative idea. Can you tell us why you chose to focus on such an everyday piece of litter and what sort of toxins and pollutants are contained in cigarette butts?
Yeah, sure. So the idea comes, I was in a conference like before we start with this project, and I saw a study about the effect of cigarette butts on other organisms. And I have to admit, I never thought about that before. I am a non-smoker, so when I saw it, I was like, aha, okay, that's also something very interesting since you can see them around you every single day. So you are waiting for the bus and you can see them there. You are waiting for the S-Bahn and you can see them there. You can even see your friends. You have seen your friends throwing them to the street. So that was quite interesting for me. And I decided like, I would really like to do a study where we focus on the effect of cigarette butts and all the chemicals that they can release with the system that we work. And that's what I come with the idea. And then Nele came, and then I suggested her, like, what do you think? I suggested her, without trying to be very biased to that idea that was really there in my head, I suggested her several topics and I let her decide. But then maybe Nele can tell us why she was also on board with this topic.
For me, it was exactly the same thing as you said, Erica. I've never thought about this as a research topic. And I think I don't know if I hesitated in that moment, but you told me the different topics. I think it was cigarette butts and then some metals or some chemicals that you also, and I was like, no, it's cigarette butts, obviously. So I was very excited when you proposed that, and then I just looked up papers, what I could find, what they were looking at. And I thought, this is crazy. I mean, there is research out there, and it's not like a lot compared to, I don't know, research about microplastics or nanoplastics, but there is a lot of research out there, and you can um, find information on the effects of cigarette butts on different organisms. And I think um, it's super interesting because when we walk through the city by a lake or a river, anywhere really, in most places we will see cigarette butts, as you said, Erica, but we don't really see them anymore. Like we don't really, realize, oh, there's a cigarette butt lying around there. It's just in the picture of the city. They're just there, and it's so normal we don't register them too much. At least that was my experience. And to me, that's what makes them so dangerous because we don't necessarily register them as quote unquote proper trash or proper trash item or pollutant. And plastic is a pollutant, right? Pesticides are industrial waste, sewage, and so on. They're discarded in the environment and they're obviously not supposed to be there. And there's so much research that really illustrates and reports on the devastating effects of those. People are really like made aware of them being a problem, and we don't see that with cigarette butts too much yet. I think they haven't quite gotten that kind of attention yet, especially in the public eye. And it's completely normal to dump your cigarettes wherever you just smoke them without actually discarding them into a trash can. And I think this is really interesting, like that there's not too much light being shown into that like research gap yet. And that's why I thought would be interesting to dabble into that. Yeah. And obviously, with all of the research out there, there's different approaches. You have studies looking at self-rolled cigarettes. You have studies looking at industrially, I don't know how you say that,
Oh,
store-bought
well,
cigarettes.
we would call them in the UK on the street, you'd call them straight cigarettes rather than rolled cigarettes, but I don't actually know if that's the technical term. Not very scientific.
let's just take
But
that.
we know what we said.
Yeah.
We know what we said. So straight cigarettes, there is studies that have had people smoke them for them. There's studies that had machines smoke, like simulate smoking behavior and had the machines smoke the cigarettes. Different tobacco types, different filter types, different paper types. And I think we decided to go with self-rolled cigarettes because we could be really sure that we could control what was going into the cigarette. Otherwise, you have to check what this company uses these kind of filters, and another company might use other filters for the straight cigarettes. So we thought this would be a great approach. And we had a lot of people at the institute that we knew self-rolled their cigarettes. So that was perfect.
Added value.
And that's added value. And that's why we decided to go with self-rolled cigarettes where we could note down the brand of the tobacco, of the filters, filter size, what was in the filter, and papers as well. Paper brand, what was in the papers, and so on.
Nice. Yeah. It seems like two things struck me when I was reading the paper, that it's a really good idea, first off, but also that you're doing this research in Berlin, which I have to say, as someone who's used to live in Berlin, is one of the smokiest cities I've ever been to. So it's really, really applicable, I think, to Berlin, because so many smoky bars and lots of smoking culture, I would say. And secondly, obviously, that when you look at other types of litter, let's say, crisp packets or, just general waste, food packets, stuff like that, that litter isn't really designed to absorb toxins or sort of have this almost biological role when you're smoking. It takes in all the toxins. It has a really high surface to area ratio, lots of fibers in there to try and absorb the toxins from the cigarette and not let them get into your lungs. Well, what other type of litter that we use every day has that? Basically nothing. So it's a really good example of, not just a piece of litter, but also almost a proxy for pollution, I would say. So that's what really struck me when I was reading it and why I really enjoyed the paper. I think we all seem to have had that revelation at the same time that you were talking about, that, oh, wow, yeah, these things are everywhere and they could be really bad for the environment.
Totally. I think, as you said, it's something that, now you said it, and also Nele was commented before, it's something that we don't really consider as a pollutant. Actually, there has been some studies where they asked the people why they throw away the cigarette butts. And some people said because they are biodegradable, it's not a big deal. But that's not true. So cigarette butts, as you said, they are like sponges. Of course, they are done for that. That's the purpose of the cigarette filter, to try to filter, to retain some of these pollutants. But the problem of the cigarette butts are not only, let's say, the cigarette butt itself, but rather that they can accumulate and later release a lot of chemicals. For instance, the cigarette butt can have a very high concentration of metals that are, some of them can be toxic for us humans and also for other organisms like copper, iron, or zinc. Also, cigarette butts can have high concentrations of nicotine, which is the main found in tobacco. And we most likely have heard a lot about nicotine because it's a stimulant compound, but also can unfortunately be very addictive. So actually, this is the molecule that it's like, let's say, the main molecule related with cigarettes. And also, these cigarette butts can contain polycyclic aromatic hydrocarbons, which are very complex chemical compounds. But in simple words, these are compounds that are going to be produced through the burning of the tobacco. So they can contain all these chemicals. And on top of that, the filters are made out of plastics. So they can release tons of fibers that are also let's say, they are also participating in the contamination of in the plastic sphere, let's say. So they are not only releasing chemicals, but they are also contributing with plastics, microplastics into the environment. So they are indeed quite a big problem.
Yeah, indeed. And as I said, an amazing piece of research. Really enjoyed reading this paper. In fact, it was actually brought up in a parasite discussion club that I'm a part of, where we just look at papers at random, and I was like, oh, I recognize those names, and I really like this research. Let's get in touch. So we've talked about cyanobacteria and cigarette butts individually, but can you summarize for the listeners what effect cigarette butts had on the cyanobacteria, and how did it affect the dynamics in this freshwater ecosystem?
So we had hypothesized that the cigarette butt solution, so the cigarette butt water that we had prepared beforehand, essentially just soaking the cigarette butts in water before getting started, distilled clean water. And we had exposed the cyanobacteria individually, but then also like separately, but then also the cyanobacteria with the chytrids together. So we had hypothesized that the cigarette butt liquid inhibits the cyanobacterial growth, and our results partially supported that hypothesis. We had also hypothesized that the cigarette butt liquid would negatively impact the chytrids, and by that disrupt the host parasite dynamics that we were looking at. And this may be a good place to mention the parasite that we were looking at, which were the chytrids, Chytriomycota, and short chytrids. Chytrids are aquatic fungi that infect cyanobacteria and have been shown to play a role in controlling or declining toxic blooms. They have a super simple life cycle in which a flagellated zoospore, so a zoospore with a tail, infects, in this case, the bacterium, so our cyanobacterium, and the zoospore will grow into a large ball after having infected that bacterium, and then will grow into that large ball, which is called a sporangium, and in this sporangium, new zoospores will be produced. And when enough zoospores are produced, the sporangium will burst, and then the zoospores will be released into the environment or into the medium, and then these zoospores can go on and infect more bacteria. So
Nice.
that's like
This is a super interesting parasite that actually we've talked about a little bit more on this podcast in previous episodes. But it's a really good model system, right? To sort of study host parasite dynamics and the ecosystem functionality of parasites, and bringing that into the experiment alongside cyanobacteria is very interesting to me as someone who studies parasites as well.
absolutely. It's also a very beautiful parasite just to look at.
Oh, really?
Yeah, absolutely. I cannot tell you how enjoyable it was to find a really nice mature sporangium. Erika knows about this. She's
Well,
experienced
that
it
sounds
many
like a
times, and
pleasure I have
it
never
is.
experienced.
It's beautiful, Joe, I'm telling you.
Well, I have to be a little bit skeptical because I think that all biologists think that their study species is beautiful. You know, like even me,
Sure.
when I started with Daphnia, I thought they were quite ugly. Now I think they're very cute. So yeah, I'll take that with a pinch of salt.
Please do.
You
Please
know,
do.
in our case, I can be sure that we have one of the most beautiful systems. I will totally agree with Nele. I mean, come on, cyanobacteria, colorful, I mean, beautiful. And then you can see that sporangia that, of course, if you think about it, it's quite creepy because it's sucking the life of the cyanobacterium. But it's beautiful to see it growing and then to see the zoospores inside. So I agree with Nele. We have one of the most beautiful systems. But okay,
Okay, when I'm
we
wrong,
can
I'm
discuss it
wrong.
later.
Yeah.
Yeah.
Getting back to the results or what we find in the study, cyanobacterial growth in the infected cultures remained relatively unaffected, and the biomass in those infected cultures under exposure to the cigarette butt liquid actually reached levels comparable to uninfected control cultures. So we basically we saw wonderful growth in infected cultures, which, if you think about it, should not be happening because the parasite is there, and that should have a negative effect on the growth. That being said, this is really cool. seeing that the cyanobacterial growth was this unaffected, because this suggests that the presence of the parasite reduces the impact of the chemical mixture on the host. So cyanobacteria kind of thrive at the expense of the parasite, and this would mean that the presence of the chytrid parasites might, to some degree, benefit cyanobacteria in polluted environments.
Well, that's a really good takeaway that this pollution can sort of affect one side more than the other. That's what it sounds
Yes.
like, that you are basically showing that the parasite took more of the damage from this pollution than the cyanobacteria, which is very interesting. I guess maybe this is to do with the sort of different cellular biology of these two species, that funguses and cyanobacteria are very different. Maybe they have different levels of susceptibility.
Could be, yeah. But that's not all. So in the uninfected cultures exposed to the cigarette butt liquid, cyanobacterial growth was hindered. So when the parasite is absent, this like reduced inhibitory effect of the cigarette butt liquid that we saw in the infected cultures is lost. So to just put it in one sentence, the presence of the chytrids in the cigarette butt exposed cultures benefits cyanobacterial growth. So we have more cyanobiomass. And when the chytrids are absent in cigarette butt exposed cultures, cyanobacterial biomass is lower. So it actually really shows that the presence of the parasite is a key component. So it's really about the interaction. So we've talked about the chytrids briefly in being a factor in lowering toxic cyanobacterial blooms. And if the host parasite dynamics are disrupted, essentially the fungus cannot perform in that lowering or quote unquote controlling role anymore. It can't perform as it should.
Wow,
in
interesting.
the environment,
Yeah.
essentially. Yeah.
It seems like there's a preferential absorbance rate, I guess, for the toxins to be absorbed more by the fungus than the cyanobacteria. That's very interesting. It's making me think what the long-term effects of this sort of pollution could be on aquatic ecosystems, in the environment, and how this might disrupt the balance of cyanobacterial and algal populations in, let's say, urban freshwater environments where you're more likely to see more pollution from cigarette butts.
Yeah, I would just want to make a comment from what you were mentioning before about the absorbance. We cannot really say that chytrids are absorbing more. We cannot really say that. So what's the mechanism behind of the results that we observe? It's really something that I'm still studying. This is something that we try to understand, but with the results that we have from this paper, we cannot really point out to which mechanism it's being followed to explain the results that we found. So as Nele mentioned, the most important for us is to see that the presence of the parasite is really unexpectedly benefiting the cyanobacteria. Or it's not really affecting, or it's like working like a shield. But how or why? This we don't know, and we need to try to dig a bit more into in order to understand it. But now that you mentioned all these things of the long-term effects, well, this is a bit going to these kind of cascade effects that can produce aquatic pollution, right? So if we remember what Nele was mentioning about the, let's say, the function that the chytrids have on cyanobacteria and mainly cyanobacterial blooms, you are expecting that these chytrids, together with other environmental factors, right? But in this case, we are going to focus on these two. If you have the chytrids, you expect that they can control cyanobacterial growth. So they always need to be in balance, right? This is life usually. You need to have a balance. So chytrids are going to kill, or they are going to suppress some growth of cyanobacteria, then cyanobacteria can grow again, and so on. So they are always in balance.
Like the classic prey and predator sort of boom and bust cycles that you see in more relatable subjects, let's say.
Exactly, totally. So you will have all the time these like one like growing more, and then the other one attacking, and then again reducing the I don't know, the number of cells of the other one. So they are always more or less chasing each other, let's call it like that. But when you break this equilibrium, when this dynamic is affected or negatively affected, and the chytrid cannot control anymore the growth of the cyanobacteria, you could expect that the cyanobacteria can overgrow again, and this can potentially lead to the formation of blooms, because we observe that it's when they are there together, well, the worst part is taken by the chytrid. So the cyanobacteria, even though it's not growing, let's say, happily, at least it's growing.
Mm-hmm.
So this is a problem, and this is something that can or suggest that the pollution with the chemicals released by the cigarette butts, as well as other pollutants, can inhibit the infection of the chytrids, which are one of the main control mechanisms for cyanobacteria to grow, and this can trigger the formation of blooms, in like really looking forward to a cascade effect, right?
Not a great prediction for our urban waterways, but definitely something that needs to be talked about and something we need to maybe look to legislate against. These cigarette butts, again, they're so casually tossed aside, but they can have real effects on people's drinking water, bathing water. Yeah, very interesting. And it's really opened my eyes every time I learn something new in science like this. I think there's another thing to worry about, but unfortunately, it's an important part of being a scientist.
Totally.
Now, this research we've just discussed focuses more on the ecological effects of pollution on cyanobacteria and chytrid populations, but are you working on any other projects that address how pollutants such as these cause negative effects at a cellular level? Maybe this is more aimed at Erika, given her current position in research, but it would be very interesting to know the specific effects, inside the cell, as well as just the ecological effects.
Yeah, totally. So yeah, I am working with other projects. So I also have studied, using this system, the effect of a herbicide, which is called metolachlor. And what we observed is that in a long-term exposure, that means for longer times, if you expose them for longer times to this herbicide, the chytrids are damaged, let's say. They have the herbicide can produce a negative effect on the chytrids. So it's not the same effect as we observe with the cigarette butts. Let's say this is more or less that they are working as a shield for the cyanobacteria. With the herbicide, it's not the same effect, but we can also see a negative effect on the chytrids, which in a long term can produce that they are not as effective as they were before the exposure to this pollutant. So they cannot control as well the growth of the cyanobacteria. And this effect that I observed is not something isolated. So the effect that we have with the work of the cigarette butts with Nele, the effect that we observe with the metolachlor, it has also been shown for other pollutants like plastics and other herbicides. And this together can tell us that, of course, there is more or less like a trend. Pollutants can damage this interaction, these dynamics. So this is one part that I have done. I am trying to understand the why, what's going on. So trying to understand a bit more the mechanism, how these pollutants released by the cigarette butts are affecting the cyanobacteria, and also to try to understand the chytrid part. How are they affecting the chytrids? So what I am trying to study at the moment is to see how or which specific mechanisms inside the cells are the ones that are damaged or responding to the stress levels. But I am not only studying, let's say, what is the stress level that these organisms have, but also going a bit deeper, trying to do a transcriptomic analysis. And I know that this sounds a bit like, what the hell is that? But transcriptomic, let's try to think about our DNA, all the DNA that we have in the cells, the same as cyanobacteria and chytrids. It's a massive cookbook, right? So they contain the recipes, which are the genes, to produce everything that the cells need. The transcriptomic is like taking this recipe book and take some copies of the recipes that at the moment are in use. And these recipes are the ones that I am trying to understand at this level, at the level of the genes. Which genes are, let's say, used or not used when they are exposed to the pollutant. So I'm trying to understand a bit more this mechanism.
Interesting. So to use this cookbook metaphor, the effects of the pollution could be highlighting or crossing out different parts of the cookbook and promoting certain recipes
Exactly.
and reducing others. Is that right?
Exactly. It's like having your recipes and suddenly saying, I want to prepare the cake, but oops, now I have the pollutant and now I don't know how many butter I need to add. So you are going
Okay.
to have this kind, as you said, like crossing or saying, maybe too much butter. Instead of 1 gram, let's put,
Mhm.
I don't know, 4 cups of butter. So this is what I am trying to understand
I
at the
see.
moment.
Okay. Yeah. Yeah. And as I said, I don't have too much knowledge of transcriptomics, but I really appreciated that metaphor. Uh, but yeah, I guess with the herbicide stuff, the cellular mechanism might be a little bit more easy to understand just because of the purity of what's going in. With the cigarettes, I saw in the paper there was so many different toxins that were listed. So I guess that trying to understand the effect of each different, as you said, metals or cyclic hydrocarbons, I can't quite remember the name of it, but all of these different, chemicals could be having different effects on different cellular processes and different parts of the parasite and different parts of the cyanobacteria. So a lot to get your head around, I understand.
Totally. I mean, that's the, let's say that's the two level of complexity, right? That we
Hmm.
have in the one hand, or on the one hand, we have this herbicide, which is only one compound that we more or less know what is going to, or we can try to understand what is like doing this specific compound. In the case of the cigarette butt solution, you have thousands of chemicals. So of
Mhm.
course, it would be very difficult for us to point out a specific or to try to relate a specific effect with a specific contaminant or pollutant. But the idea of having these kind of solutions is not really to understand what each component is producing, but rather to understand this solution as a whole, what is the effect that it's having. Because at the end, in the nature, we have this complex mixture of chemicals. We not have only one pollutant and then remove that one and arrive another one. No, in reality, we have all these complex mixture. So that's the reason why this study with the cigarette butts is so interesting, because that can also allow us to see a bit more how this complex chemical solution, mimicking the complex chemical solution that we have in the nature, can also like affect organisms once in a while, like all together.
Yeah, you've got to keep it biologically relevant, I guess. But at least with all these complicated interactions, there's so much more research to be done. So I guess that's the good side of things, at least.
Totally. We still have a lot of things to do.
Yeah. So finally, I mean, we've talked a little bit about cigarette butts already, but I'm sure that everyone listening agrees that these are not in short supply. We probably see hundreds of them, every week on the floor and in rivers, sadly, and in environments. But I have to say that I'm quite curious about how you managed to get them for the research, because you also mentioned that it was someone or some people that you were working with that used the rolled ones. But it still seems like a slightly weird question to go up to someone and ask them for their butt of their cigarette and that you need it for quote unquote research. Did you feel a little bit silly doing that when you were approaching, hopefully not strangers on the street?
Maybe a little, but it wasn't too bad because I mean I'm kind of taking the magic out of this now, but we were asking people at the institute. So they're exposed to science every day. So if someone comes up and is like, I need this and that from you and it's for research,
Hmm.
then
No
it's
questions
probably
asked.
not too, no questions asked. Here's all my cigarette butts. Please take them. But I think we did make a little bit of a joke out of it. So for example, in the presentation for my master thesis that I gave at the very end, I think I photoshopped like a picture of people on a beach, like really shoveling cigarette butts into like a blue trash bag. And I put like Erica's and my face over it. So people were kind of, I was like, yeah, this is actual footage of us getting these cigarette butts. but when in reality it was a little bit less embarrassing and a little very simple for us because we just had to ask, and then people were like, yeah, sure.
Way
No
better
problem.
than going out on a cold lake and taking samples. So yeah, I understand that.
Totally. However, I mean, I just have to add to what Nele mentioned. I did ask to some friends outside the institute.
Oh, you did?
And some of some of the cigarette butts that we have were from some friends outside the institute, and they were like, but why? So they were really wondering, why do we want to have that? And they were like, but this is very disgusting. And I was like, yeah, but we need it. We need it. And one story that is very funny. I don't know if I never told you, Nele, but I mean, here where I live, I went to some of my neighbors because all the time when I'm coming home, I can smell that someone is smoking. So I thought it was my neighbors close by. So I went, and you can imagine, like, hi, I am blah, blah, blah. So because we have never spoken beyond the hello, choose, so the normal one. So I just went and I was like, hi, I am Erika Martinez. I am blah, blah, blah. So I tell them where I'm working and that I needed that if they don't have like some cigarette butts. And I remember that she was, because it's a she, so she was like, oh no, I'm very sorry, I don't smoke anymore, but if you need it, I can start smoking again. And I was like, no, we don't want anybody to start smoking again because of us. And she was like, but seriously, I can't do it. And I was like, no, thank you, really don't. So because it was very funny. But as Nele mentioned, the majority of the cigarette butts, we got it from people at the institute. I mean, we are not going to say names, but we have one of our best donors, let's say, that gave us more like almost half of the cigarette butts, right, that we got. But luckily they were like, yeah, why not? So that makes me wonder how or which other things they have been asked to do, which other weird things they have been cooperating with. But for us it was quite funny to get all the cigarette butts.
Well, that sounds like you have very helpful neighbors, at least, that you can count on in the future. And I think with things like this, right, it's just like if someone comes and asks you for something like cigarette butts, you assume it's either art or science, and you're just like, okay, you just do you.
Yeah. They are very supportive, as you said.
Yeah, that's very nice indeed, but still funny. Great. Well, I think that's all the questions that I have then, and I've really enjoyed speaking to you about this really fascinating paper. I'll make sure to put the title and the reference number in the episode notes so anyone who wants to read it can. Thanks again for joining me on this podcast. I hope you guys have had a really good time, and it's been really interesting to talk to both of you at the same time, podcast first, and I think it went pretty well. So all that's left for me to say is thank you very much, both of you.
Thank you. Thank you for having us. Thank you for talking to us.
Yeah, thank you very much, Joe. It was a nice experience.
The ParAqua podcast was funded by COST, the European Cooperation in Science and Technology, and is a product of the ParAqua Action Group. If you are interested in joining an action group or just want to know more, please go to www.paraqua-cost.eu or follow the link in the episode description. The podcast is produced by myself and Serena Rasconi, and the Science Communication Coordinator is Anna Gavrilovich. Once again, thanks for listening, and see you next time on the ParAqua podcast.